have discussed xylem flowing, we have discussed this. Okay. Then there is root. There is structure of root. Um, you should know the structure because it was in the past paper. I don't think that they're going to give this question again, but it was related to, okay, the diagrams looks like this. Okay, wait. No, structure, internal structure of root. The diagram is like this. Okay, one is for the dicotyledon, other is for the monocotyledon. Okay, what is the difference between dicot and monocot? Exactly, two cotyledons. We don't say two seeds, we say two cotyledons. Two cotyledons. Okay, what else? Yes, what cotyledon in the seed? Okay, what else? The network of veins and good. Three and multiple of three in mono or multiple the petals. Okay, what about the veins? The direction of veins. Network. Okay, so we don't say like this, we say parallel. Okay, the word is parallel veins. We need to remember this word. Okay, there was question related to it. They have given different type of plants, and then they ask that what is present in that. Okay, you have to select the you have to select the plants for dicotyledon. Okay, there was one MCQ related to that. Okay, let me check if I have that question right now. I'll share. Okay, there were different leaves given, leaves and plants. Oh, sorry, flowers. The leaves were given like this, and there were straight lines. Then this was given with the different petals, and then they tell select the plants which are dicotyledon or which are monocotyledon. Okay, yes, you need to look at the number of petals and the direction of veins, if it's parallel or branched. Okay, okay, okay. This diagram you must know. One is related to dicotyledon, other is related to monocotyledon. Okay, here in root, wait, the upper one. Yes, we need to. No, we don't have to memorize this word. We just have to memorize the position of xylem and phloem. Okay, only the position of xylem and phloem. Keep in mind that unlike the stem, in stem, the xylem is present outside. Oh, sorry, xylem is present inside and phloem is present outside. Okay. Similar, phloem is present outside, xylem is present inside. Here also xylem present outside, xylem present inside and phloem present outside. Okay. This is the only thing that you need to learn, their position. What are you humming? Hmm. Okay. No, cortex is the one which makes the wood. Okay, the which makes the like for example, if there are root hair cells present here, okay, then the part next to it is the cortex, which contain number of cells. These are the number of cells which increases the width of stem or width of root. Okay. um where was the question? Okay, I will give you one file for MCQs. You need to learn that. Okay. Okay, right now we will be discussing the limiting reactants. Let's just, okay. I have a question for you. Wait. Let's copy this question because this one is important. Okay, we have an experiment because this chapter is really important with respect to ATP as well. Okay, first of all, the question is state the balanced chemical equation. Hmm. Done. Okay, done. So there are 3 to 4 equations that you must learn. Okay. But 2 is present over here. Okay. Now this one is related to the investigation of the aquatic plant, the water plant. Okay. An experiment or has been designed, or the apparatus has been set up. There, this apparatus is used. Okay. Let's see why they have used syringe to collect oxygen gas. Okay. You must know this. Why is syringe used over here? Okay. Why do we use sodium hydrogen carbonate for aquatic plants? As a source of carbon dioxide, because it has no contact with the air, so it must get carbon dioxide for photosynthesis. It would get it from sodium hydrogen carbonate. Okay, why do we use lamp? Lamp light. Okay, and why are we using this thermometer? The temperature. Okay. Okay, one thing I want to ask, is photosynthesis an endothermic process or exothermic process? Yes, because the temperature decreases in this one. Okay, I'm giving you one question. Just go through this question because it's related to practical as well. State why the student keep the lamp at the same distance during the investigation. Okay, wait. Let me give you the whole statement. The question is about a student investigated the effect of different wavelengths. Okay, on the rate of photosynthesis. So, what is dependent variable and what is independent variable? Exactly. Wait. What are we investigating? The effect of? No, we are investigating the effect of wavelength. So this would be independent variable. Not the lamp, but the type of light we are using. Yes. Because the next question is related to lamp. Okay. The next thing is independent variable would definitely be the number of oxygen bubbles produced. How would the volume of gas collected? Okay. Volume of gas collected or number of oxygen bubbles. Let's say. Because we are using syringe over here, so we have to. Hmm. Exactly. Exactly. We have to take we have to write the same term as they have used in the question. We cannot change the terms. Okay. Okay, next thing is keep the lamp at the same distance during the investigation. Why should we keep the distance same? Same distance. Why should the distance be same? Yes. No, we are on the same experiment. We are changing only the type of light, not the light intensity. So the light intensity would be constant. Okay. There are two things related to light. One is wavelength, other is intensity. Okay, wavelength depend on color, whether you're using blue color, green color, yellow color, red color. Okay. Intensity depends upon the distance. Like the light intensity is very high at the afternoon, but very low in the early morning or in the evening. Okay. So if we are investigating the type of light, then the other factors should be constant. So our distance should be constant so that the light intensity is constant. We cannot change it. Otherwise, this factor would also become the independent one. We have to keep one factor independent only. There's no need to have multiple independent factors. Okay, the variables. Okay, why do we use sodium hydrogen carbonate? to provide carbon dioxide for plant. Okay. State 3 uses in a plant that carbohydrate produce in the process. Let's talk about the uses of carbohydrates that are produced. Energy, good. By aerobic respiration. Okay. In making chlorophyll. Okay. I'm telling you there's six uses, just write it. Okay. They are important in making proteins with nitrates. Carbohydrates plus nitrate is equal to protein. Okay. Then they are helpful in making the nectar, helpful in storing the plant in storing the carbohydrate in the form of starch, and then they transport it in the form of sucrose. I have one more question for you now related to sucrose and starch. Okay, we say plant make glucose, transport sucrose, but store starch. Why why there are three type of carbons? Starch and the one which it makes, glucose. Mhm. Okay. Why they're important for plants? Hmm. Wait, I'll tell you the answer. Okay. Glucose and sucrose are simple sugars. Okay. They are soluble in water. Let's say water soluble. And the third thing is they can change the osmotic balance. Okay, what is osmotic balance? Now, if there are more salt, if there is more salt, it would become hypertonic. If there is less salt, it would become hypotonic. Okay. So if the sucrose is present in very high amount in a plant, then the plant would suffer from the plasmolysis. They can change the osmotic pressure, osmotic, related to osmosis. Okay, osmotic plus. Okay, why it store as starch? Starch is a complex sugar, insoluble in water, so it won't disturb the water balance or the osmotic balance. Yes, exactly. Okay, let me explain. No, we have to use the word osmotic. Related to water. Yes, osmosis, related to osmosis. Okay, now listen. Once it is stored in plant, converted into a complex sugar, it cannot dissolve. Now there won't be extra salt in the water. It means water cannot be like reabsorbed or water cannot exit the cell. That's why in us as well, in humans, the glucose is converted into glycogen, right? If we have extra glucose, that is converted into glycogen by insulin. So if it's converting into insulin, it means it's maintaining the some balance in our body, the glucose level. Okay, what, the simple sugar one or the glycogen one? Glyco. Okay, we eat we eat sucrose or starch. Okay, then it is converted into glucose, reabsorbed, sorry, absorbed by our body. And if there is extra glucose, that is stored as glycogen. This is a function of insulin, right? I'm telling. Wait. Half of the answer you have told already. Half of the answer you have told. What have you told? That they need to be broken down into smaller pieces like glucose so that they can be reabsorbed through the microvilli, right? This is the half answer, right? This is correct. Now the next half question, that why it is converted into glycogen. because glycogen is the insoluble form. Okay, it is not going to disturb the sugar level or the glucose concentration in our blood. Okay, wait. If it's dissolved in our blood, it would definitely create a hypertonic situation. If there is a hypertonic situation, then red blood cells are going to be destroyed because the water come out of the cell. Simple. So we need to have a certain insoluble. Exactly. Exactly. Which we need to maintain. Okay. Okay. What we were discussing? Okay, why they are converted. Okay. So these are the six uses that you must know. Okay. One, two, three, 4, 5, and 6. Okay, protein with nitrates. Carbohydrates unites with nitrates to make proteins. Okay, which nutrients are absorbed by plants from the soil? Let's talk about the nutrients. The importance of nutrients. There are two nutrients important in your syllabus. One is magnesium, other is nitrate. Okay, magnesium is important for making chlorophyll. If there is no magnesium, the plant would turn yellow. Simple. There won't be photosynthesis. The plant won't grow because there is no photosynthesis. It cannot make food. It cannot produce energy. Right? The very first thing that you need to mention in magnesium is the chlorophyll, whether chlorophyll is there or not. And what would be the effect of not having a chlorophyll if it's a a 4 mark question or a 3 mark question? There's no need to mention the word flaccid. Okay, there won't be any energy, so there won't be any growth. Okay, why are nitrates important? Nitrates are important for making amino acids. Amino acids further make proteins. Now, whether it's plant or animal, protein is for growth. We need protein for growth. Plants also need. Yes, nitrates make amino acids. Nitrates, along with carbohydrates, make amino acids, which further make proteins, which are important for plant growth. Why are protein important for plant growth? Okay, proteins are involved in cell division, in development of cells, in the repair of cells, in making new cells. Oh, one function I missed. Wait, one function of carbohydrate I missed. Sorry, that is cellulose, which is important part of our of cell wall. None? Okay. Now let's have another question. I don't know where my questions are going. Okay, here. Okay, I'm giving you one more experiment. Okay, let's talk about the same experiment. Why do we use a thermometer for the temperature? And why are we using this water beaker? This one. Okay, why do we use water bath in experiments? Let's rephrase it. To heat the water. Okay, I'm giving you one graph. You need to describe the graph. There is no question related to um, the explanation. You need to describe the graph and tell what is the limiting factor at x. I'm giving you this. Wait, here is the graph. I'm sending you on in like chat as well. Here, one. Okay. So here is a graph. You need, first of all, you need to describe, and then you have to tell what is the limiting factor at X and what would be limiting factor at Y. Okay, what are the limiting factors? Let's say limiting factor is anything. Yes, yes. Think again. Think again. What is the graph about? It's related to graph. It's given in the graph. So carbon dioxide at X, at X, carbon dioxide is a limiting factor. Let's say carbon dioxide concentration. We have to write this. At Y, look, we have enough uh, carbon dioxide. So what could be less? Light. You have told it opposite in the start. X would be carbon dioxide and Y would can be light. You can also say Y can be temperature as well. But light is the most appropriate one because if the temperature is increased or decreased, then the enzyme can get denatured. Okay, so better to write light here at Y. Okay. Um, okay, do you know how to calculate the rate of photosynthesis? Yes. It would be, how do you calculate it? How do you calculate the rate of any reaction? Rate of photosynthesis, rate of respiration, or anything. Let's say rate of any reaction in bio. It's the same. It's the same. It's same. The decrease in the amount of reactant or increase in the amount of product. That over time is the rate of reaction. Decrease in the reactants, whether mass or volume. Decrease in the reactant and increase in the, no, only the either we will check the decrease or increase. We have to use one, time, or increase in product over time. There are two formula that we can use. Okay. Then there is another graph given. Okay, the graph is mostly about the rate of photosynthesis like that. These will tell you the what is the limiting reactant, what is not. Explain the graph, describe the graph. Okay, how would you explain this graph if the question is like this? How would you explain this graph? Okay. Uh, okay, wait. Before this, learn few things. In description, you need to define 3 to 4 things. Okay, the very start, initial point. Okay, very start, where it's starting, then move toward the highest point. Okay, then tell what happened after that. What happened after that highest point? Is it becoming constant or is decreasing or whatever? Okay, so these 3 points would give you your marks. Okay, now the wait. Next thing, you need to mention the units given on the x-axis, given on the y-axis. Then you have to tell whether it's decreasing, increasing, or the constant. What happened after that? Till end. Mean is starting from 0% of carbon dioxide. Then the highest is at 4%. Then after 4% of carbon dioxide, the rate becomes constant till 0.5%. So you need to give how many number of bubbles are produced at each value. Okay, like how many bubbles are there at 0.0%, how many are there at 0.4%, and how many are present in 0.5%. Okay, one more thing. If there are two readings given, for example, if there is another graph given over here, then you have to compare both of these. And if by any chance, if these two lines are overlapping, then you have to mention this overlap point also in the description, that the number of bubbles produced are same at this concentration. Okay, are you writing the points how to describe the graph? Okay. Okay, so this is all. Let's stop here and then we'll discuss more questions tomorrow. Okay. Yes, 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 you can. Mm-hmm. Hmm. What three number of the number of bubbles produced were three. Okay. Okay. Hmm. Exactly, exactly, exactly. Two, 20 at. Hmm. Good. And then what happens after that? Becomes constant. Hmm. Percent. We need to mention that. Okay, I have given you one file. You have to do 25 MCQs for this one. Okay. Okay. So we'll discuss more questions tomorrow related to this graph, because graphs are important in this one, in this chapter, along with the experiments. Okay. So take care. Allah Hafiz.